Okay, this is it. Week 3, Lesson 3, iron and copper, uptake and storage. We're going to look at iron first. We'll study siderophores, transferrin, the iron transport protein, and ferritin, the iron storage protein. And then I'll talk briefly about copper, which is less well understood but has a pretty different method of storing itself in living systems. We've already seen in previous lectures the importance of iron. Iron is a very important redox carrier. All known forms of life require iron. It's usually cycling between the iron 2 and the iron 3 redox couple, although iron 4 is known in some situations. So we have the rubridoxins, the ferridoxins, the different types of ferridoxins, the Rieske iron center. So we talked about these in the last lecture. Those are redox proteins. Cytochromes are redox proteins. And of course, heme iron in hemoglobin and myoglobin are very important for transport of oxygen in your body. Okay, if iron is so important, it must be freely available in the environment, and that isn't quite true. In the ancient anaerobic Earth, where the atmosphere was methane instead of oxygen and nitrogen, the atmosphere was very reducing, and iron 2 was the more stable form of iron. And iron 2 is pretty water soluble. It's pretty available in the environment. The one exception is probably iron sulfide, which has a low KSP, and that limited some of the availability of iron, but certainly, for the most part anyway, iron was an available species. But then, about 2.4 billion years ago, and specifically March 25th, 2.4 billion years ago, photosynthesis began. And it was cyanobacteria began undergoing photosynthesis, and it changed the atmosphere to being an oxidizing environment, and almost all life went extinct. This was a very traumatic time for living things because as the atmosphere became more oxidizing, iron 3 became the more prevalent form of iron. And iron 3 is very unavailable. It has an extremely low availability because iron hydroxide has an extremely low KSP. Rust and magnetite are solids. They're insoluble in water. And all of the iron in the environment basically precipitated out as the atmosphere became oxidizing. Life had to then evolve or die out in this highly toxic atmosphere. And evolutionary pressure led to the development of the first eukaryotes. Eukaryotes are cells that have nuclei, and the separation of the nucleus from the cell allowed for DNA repair mechanisms to evolve. It also allowed the DNA to be protected from reactive oxygen species such as peroxide and superoxide. So iron is not particularly available in the environment. It's actually considered to be the limiting factor in growth. Iron has been proposed as a treatment for global warming. As life absorbs the iron that you spray onto the ocean, then the life would take the CO2 and, so for phytoplankton, which are photosynthetic, they take CO2 and turn it into sugars. And although this has been tested, it doesn't appear to be a viable way to take CO2 out of the atmosphere. And the way bacteria solved their problem is by developing compounds that are known as siderophores, which is Greek for iron carrier. And these are small molecules that are produced by the bacteria that go out into the environment and bind tightly to iron. And then there are receptors on the cell of the bacteria that can recognize the siderophore with iron bound to it, and it will absorb that back into the cell. They're produced by bacteria under stress conditions, specifically iron stress. So if they don't have enough iron, they'll produce these siderophores. The siderophores go out into the environment, and the bacteria can absorb them back in. The important mathematical consideration here is the formation constant. This is the equilibrium constant that I taught you in Frosh Chem. Kf is the metal ligand complex concentration divided by the free metal and the free ligand concentration. And siderophores are effective because they have extremely high formation constants. In fact, the formation constant for enterobactin, which is this molecule here, is the highest formation constant known. It has a Kf of 10 to the 49th, which means that the free iron 3 concentration in equilibrium with enterobactin is 10 to the minus 35th molar. Homework 4 this week will have you investigate this formation constant in a little bit more detail. But let's look at the structure of enterobactin. It's a tris catechol, so this is a catechol group. It's a bisphenol. It's linked to this lactone ring. There are 3 of these catechol groups, and they bind to iron in a pseudo-octahedral coordination environment. So this goes out into the environment, binds to the iron, and then is absorbed back into the cell. The textbook chapter goes into a little bit more detail about some of the geometrical changes that take place as this binds iron and gets absorbed back into the cell, but that's not really essential. I just want you to understand that this coordination complex is important for binding iron. Enterobactin is found in E. coli and other organisms as well, but it's mostly found in E. coli. So enterobactin is not the only siderophore. This is a different siderophore called desferoxamine B. This is found in Streptomyces. And the binding group in this siderophore is this group known as a hydroxamic acid. And so this is a hydroxamic acid. Here's another one. Here's another one. So this also binds to iron and can be reabsorbed back into the cell after it finds iron. Interestingly, humans have developed their own siderophores, not to get iron from the environment. We obtain iron from our diet, from eating food that contains iron. And we'll talk about that on the next slide. But we have developed receptors in breast milk, so lactoferrin, and eggs also have, obviously not in mammals, but eggs contain siderophores as well. And those are I believe they're called ovoferrins, but I totally forgot right now. Anyway, these absorb iron from the environment around where a bacterial infection might take place. So in breast milk, the siderophore binds to the iron, preventing the bacteria from binding to the iron, and it serves as a naturally occurring antibiotic. Okay, so let's look at mammalian transferrin. When you eat a food that contains iron, we don't want that iron to be lost. We want to absorb that iron. And there's all sorts of machinery that's been evolved to take care of this. But we don't want iron just floating around in our bodies. Iron, too, can react with peroxides and other species present in the body to make these highly reactive oxygen species that I've already mentioned. This is the hydroxyl radical, the peroxyl radical. Those are really bad. We don't want those in the body. And so we want to absorb the iron and hold on to it tightly so that we can deliver it to where it needs to go in the cell. And the protein that has been developed to do this is called transferrin. This is a fantastic crystal structure that shows both the open form and the closed form in one molecule. The iron actually binds deep in the cavity. I'm pretty sure that the iron is in this location. I think that's the iron atom there. And it holds, when it binds to this protein, it sort of makes it turn into a clamshell, and the clamshell that was open in this side clamps down and holds onto the iron very tightly. The binding pocket for the iron in the transferrin looks something like this. So we have an iron atom, and it's bound by 2 deprotonated tyrosines, otherwise known as tyrosinates. This is a histidine, an aspartate, and this carbonate, which is actually essential for binding. The carbonate is then stabilized with a positively charged nearby arginine. And so you can see that this protein works very hard to have a variety of deprotonating sites, a charge balancing site to absorb onto the iron and make this whole thing neutral. And there's a concerted binding between carbonate and iron as it binds. And that's actually important because the carbonate helps to then release the iron when necessary. So I'll talk a little bit about the transferrin receptor in cells, and that's how you can get iron into a cell. And the textbook talks about this, and I'll just briefly mention it here. So when transferrin is absorbed into a cell, an ATP proton pump lowers the pH of the vesicle from neutral to around pH 5 or 6, and at that point this carbonate begins to get protonated and it starts to collapse this whole structure, opening it up at the lower pH. The iron is then released and it gets taken into the cell and used for whatever purpose it needs to. So as the pH lowers, you can protonate the tyrosines, you can protonate the carbonate to bicarbonate, and that disrupts the binding, opening the cluster and releasing the iron. So it's really a fantastic cooperative effect. Now, it takes a lot of energy and a lot of work to bring iron into a cell. We don't want to waste it, and so we've developed proteins called ferritin, which are iron storage proteins. These proteins, they buffer the iron availability in the organism. They do this by having a little nanocrystal of iron that is just carried around in your body. And I've read a couple places that we have approximately 3 grams of iron in our body, and 1 gram is sort of just stored in ferritin, waiting to be needed if there's an iron shortage in your diet. So that's a really interesting way to store the iron, and we'll talk about the structure of it in a second. This is an extremely conserved protein. It's essentially the same in all life forms that store iron, so from algae to humans. I've had a picture in this video that I took out, but it shows that the protein that makes ferritin is essentially identical across all sorts of different life forms. So, ferritin consists of 24 identical alpha, that should be an alpha, helical-rich subunits that self-assembles into a nanocage that has 4-fold, 3-fold, and 2-fold axes, and it turns out the whole thing has O symmetry. And here's a thinking question for you. Why does it have O symmetry and not OH symmetry? OH is the point group we normally think about for octahedral. What is it about amino acids that gives us an O symmetry? And so if you are stuck, ask me in office hours and I will help you think about this problem. So iron is stored with a simultaneous release of hydrogen peroxide and protons. These are chemicals we do not want to have a lot around, especially the peroxide. Protons we can deal with. And so it's really expensive and dangerous for the cell to store the iron. That lets you know how important it is to store iron. Peroxide is normally bad to have around. And so there's actually nearby peroxidases which are present, which reduce the peroxide down to water. And so it's an extremely expensive process to store iron in the cell. The mineralization process takes on the order of hours. And in about 2 slides, I have a really, really fantastic series of crystal structures which demonstrate the slowness and importance of the slowness of this process. Okay, this is the crystal structure of ferritin. It's actually a cartoon of the crystal structure of ferritin. And I want to show you some features of this protein. First of all, the outer diameter here is on the order of 12 nanometers. And the inner diameter here is on the order of 8 nanometers. So this is a legitimate nanocrystal. You can see in this picture the 4-fold axis, the 3-fold axis, and the 2-fold axis. And it turns out if you multiply 4 by 3 by 2, you get 24, which is the number of symmetry elements in the O point group. And so that's one way to think about symmetry in collecting this. And this is the smallest repeating element of the ferritin subunit, so it's 5 by 2.5 nanometers, and that self-assembles into this beautiful octahedral cluster. Iron is believed to enter the pore through the 3-fold channels, and I'll provide some evidence for that in a little bit, and electrons are believed to enter the pore through the 4-fold axis, enter and leave the cluster. So one piece of evidence that suggests that iron goes into the 3-fold axis is if you add denaturing agents, reagents, or protons, these alpha helices that block this pore are the first to begin to come apart. And so you could imagine a proton pump or some sort of reagent here that allows this to open up while maintaining the rest of the structure of ferritin, which is important because we've got all the iron on the inside that we don't want to release. Here's a zoom in of the 4-fold and the 3-fold axes, and these have been color-coded to show a little bit more detail about the side chains present on this pore. So on the threefold axis, these are aspartate and glutamate amino acids, and so protonating these is going to change their charge, and they're going to denature and open up, whereas the lining of the fourfold axis is leucine, and a hydrocarbon, a nonpolar, and so the electrons are more able to go through in that region of the pore. So here's a cool little graphic that I found from the protein data bank. And if you're familiar with the protein data bank, this is 4LGH, 4LPJ, and 4LYU. And you can look at these structures. I'll also provide a link on the next slide. But this is a zoom in of one small region of the ferritin structure before it has been exposed to iron. So this is known as apoferritin. There's no iron inside. But you can see some binding groups in here that are likely to bind to an iron. And then they soaked the protein in a solution of iron for 1 or 2 minutes, and we saw the bringing in some iron into the structure. So now we can see 1 iron in the cluster, and then they soaked it in the iron solution for a total of 15 minutes, and we saw even more iron getting incorporated. And so at this point, there are 5 iron atoms present in each subunit of the cluster. You can see some of the iron atoms in other clusters sort of behind. And so this protein now has 24 subunits, each with 5 iron, so quite a bit of iron after only 15 minutes. The iron that gets incorporated into ferritin is then oxidized up to iron 3, and it mineralizes into, is a similar mineral to one that's called ferrihydrite, which is an iron 3 mineral that includes phosphate, hydroxide, and oxide ligands. It's all iron 3. And to release the iron back into the environment, into the cell, it has to be reduced back to iron 2, which is then soluble. And so those electrons have to come in through those fourfold pores to reduce the irons one at a time and release them back as iron 2. So a very complicated machinery to understand the binding of iron in this system. I'll talk a bit less about copper uptake. It is not as well understood, although this example of the CCS protein, which is the copper chaperone protein, and this protein delivers a copper to copper zinc SOD, which is a superoxide dismutase protein. It's a protein that takes superoxide, O2 minus, and disproportionates it into hydrogen peroxide and dioxygen. So, how does this copper protein work? First of all, remember that modern electron transport proteins, ones that have evolved since the cyanobacteria event, mostly use copper. Copper became more available than iron after the oxidation event. And so this is a redox protein. Copper is a redox element in biological system. And so the delivery of copper to the SOD protein is reasonably well understood. This is a zinc atom. This SOD has been modified to only have the zinc binding site. There is no copper binding site, but if there was, it would be right nearby here. And so they can crystallize this in an inactive form and see what it looks like. So the copper chaperone protein has 3 domains. Domain 1 here in this magenta color includes the copper binding region. So normally there would be a copper bound in that part of the protein. Domain 3, this gold color, is also contains a copper binding region. This is a cysteine and a cysteine. And there's no copper in here right now, but what happens is when the CCS protein binds to the SOD, and that takes place in this blue binding domain. So the blue part binds to the SOD. The domain 1 has a copper in it, and the domain 3 actually swings over, grabs the copper, swings back, and puts the copper into the SOD. So this random coil, this spaghetti strand here, is highly flexible and allows this binding region to swing back and forth and transport the copper. So, the take-home messages from today's module include the following. The first is that iron is really important, and we have evolved a lot of specialized proteins to incorporate iron into our bodies. In bacteria, these are siderophores, whereas in higher forms of life, these include the transferrin and ferritin molecules for transport and storage of iron. We've also seen one example of a copper uptake protein.